In this part of the test, there are two texts about different aspects of healthcare. For questions 7 to 22, choose the answer A, B, C, or D, which you think fits best according to the text. Text 1, Sleep deprivation. Millions of people who suffer sleep problems also suffer myriad health burdens. In addition to emotional distress and cognitive impairments, these can include high blood pressure, obesity, and metabolic syndrome. In the studies we've done, almost every variable we measured was affected. There's not a system in the body that's not affected by sleep, says University of Chicago sleep researcher Eve Van Cotter. Every time we sleep deprive ourselves, things go wrong. A common refrain among sleep scientists about 2 decades ago was that sleep was performed by the brain in the interest of the brain. That wasn't a fully elaborated theory, but it wasn't wrong. Numerous recent studies have hinted at the purpose of sleep by confirming that neurological function and cognition are messed up during sleep loss, with the patient's reaction time, mood, and judgment all suffering if they are kept awake too long. In 1997, Bob McCarley and colleagues at Harvard Medical School found that when they kept cats awake by playing with them, a compound known as adenosine increased in the basal forebrain as the sleepy felines stayed up longer, and slowly returned to normal levels when they were later allowed to sleep. McCarley's team also found that administering adenosine to the basal forebrain acted as a sedative, putting animals to sleep. It should come as no surprise then that caffeine, which blocks adenosine's receptor, keeps us awake. Teaming up with Bashir and others, McCarley later discovered that, as adenosine levels rise during sleep deprivation, so do concentrations of adenosine receptors, magnifying the molecule's sleep-inducing effect. The brain has cleverly designed a two-stage defense against the consequences of sleep loss, McCarley says. Adenosine may underlie some of the cognitive deficits that result from sleep loss. McCarley and colleagues found that infusing adenosine into rats' basal forebrain impaired their performance on an attention test, similar to that seen in sleep-deprived humans. But adenosine levels are by no means the be-all and end-all of sleep deprivation's effects on the brain or the body. Over a century of sleep research has revealed numerous undesirable outcomes from staying awake too long. In 1999, Van Cotter and colleagues had 11 men sleep in the university lab. For 3 nights they spent 8 hours in bed, then for 6 nights they were allowed only 4 hours, accruing what Van Cotter calls a sleep debt, and then for 6 nights they could sleep for up to 12 hours, sleep recovery. During sleep debt and recovery, researchers gave the participants a glucose tolerance test and found striking differences. While sleep deprived, the men's glucose metabolism resembled a pre-diabetic state. We knew it would be affected, says Van Cotter. The big surprise was the effect being much greater than we thought. Subsequent studies also found insulin resistance increased during bouts of sleep restriction, and in 2012, Van Cotter's team observed impairments in insulin signaling in subjects' fat cells. Another recent study showed that sleep-restricted people will add 300 calories to their daily diet. Echoing Van Cotter's results, Bashir has found evidence that enforced lack of sleep sends the brain into a catabolic, or energy-consuming, state. This is because it degrades the energy molecule adenosine triphosphate, ATP, to produce adenosine monophosphate, and this results in the activation of AMP kinase, an enzyme that boosts fat synthesis and glucose utilization. The system sends a message that there's a need for more energy, Bashir says. Whether this is indeed the mechanism underlying late-night binge eating is still speculative. Within the brain, scientists have glimpsed signs of physical damage from sleep loss, and the timeline for recovery, if any occurs, is unknown. Chiara Sorelli's team at the Madison School of Medicine in the USA found structural changes in the cortical neurons of mice when the animals are kept awake for long periods. Specifically, Sorelli's colleagues saw signs of mitochondrial activation, which makes sense, as neurons need more energy to stay awake, she says, as well as unexpected changes, such as undigested cellular debris, signs of cellular aging that are unusual in the neurons of young, healthy mice. The number of debris granules was small, but it's worrisome because it's only 4 to 5 days of sleep deprivation, says Sorelli. After 36 hours of sleep recovery, a period during which she expected normalcy to resume, those changes remained. Further insights could be drawn from the study of shift workers and insomniacs, who serve as natural experiments on how the human body reacts to losing out on such a basic life need for chronic periods. But with so much of our physiology affected, an effective therapy minus other than sleep itself is hard to imagine. People like to define a clear pathway of action for health conditions, says Van Cotter. With sleep deprivation, everything you measure is affected and interacts synergistically to produce the effect. Text 1, questions 7 to 14. 7. In the first paragraph, the writer uses Eve Van Cotter's words to A. Explain the main causes of sleep deprivation. B. Reinforce a view about the impact of sleep deprivation. C. Question some research findings about sleep deprivation. D. Describe the challenges involved in sleep deprivation research. 8. What do we learn about sleep in the second paragraph? A. Scientific opinion about its function has changed in recent years. B. There is now more controversy about it than there was in the past. C. Researchers have tended to confirm earlier ideas about its purpose. D. Studies undertaken in the past have formed the basis of current research. 9. What particularly impressed Bob McCarley of Harvard Medical School? A. The effectiveness of adenosine as a sedative. B. The influence of caffeine on adenosine receptors. C. The simultaneous production of adenosine and adenosine receptors. D. The extent to which adenosine levels fall when subjects are allowed to sleep. 10. In the third paragraph, what idea is emphasized by the phrase by no means the be-all and end-all? A. Sleep deprivation has consequences beyond its impact on adenosine levels. B. Adenosine levels are a significant factor in situations other than sleep deprivation. C. The role of adenosine as a response to sleep deprivation is not yet fully understood. D. The importance of the link between sleep deprivation and adenosine should not be underestimated. 11. What was about the findings in Van Cotter's experiment? A. The rate at which the sleep-deprived men entered a pre-diabetic state. B. The fact that sleep deprivation had an influence on the men's glucose levels. C. The differences between individual men with regard to their glucose tolerance. D. The extent of the contrast in the men's metabolic states between sleep debt and recovery. 12. In the fifth paragraph, what does the word "it" refer to? A. An enzyme. B. New evidence. C. A catabolic state. D. Enforced lack of sleep. 13. What aspect of her findings surprised Chiara Sorelli? A. There was no reversal of a certain effect of sleep deprivation. B. The cortical neurons of the mice underwent structural changes. C. There was evidence of an increased need for energy in the brains of the mice. D. Neurological response to sleep deprivation only took a few hours to become apparent. 14. In the final paragraph, the quote from Ben Cotter is used to suggest that a. The goals of sleep deprivation research are sometimes unclear. b. It could be difficult to develop any treatment for sleep deprivation. c. Opinions about the best way to deal with sleep deprivation are divided. d. There is still a great deal to be learnt about the effects of sleep deprivation.